My name is Shruti Benny. I am from 3rd BSc Microbiology and my topic is Treponema pallidum. First of all, coming to the history and morphology. It was discovered by Fritz Schaudin and Paul E. Hoffman in the year 1905. It was discovered in the serum sample. Genome of the particular bacteria was discovered in the year 1998. The word treponema was coined out from two words, tripos and nema. Tripos meaning to turn and nema meaning thread. The organism is thin, delicate, gram-negative spirochete and it has tapering ends. The size is, it would be having 10 μm in length and 0.1 to 0.2 μm in width. It is a motile organism and it would be exhibiting three kinds of movements. First of all, it exhibits rotation around the long axis. Then it would be having backward and forward movements and the whole body would be flexible. The cell also has a central protoplasmic cylinder which is bounded or covered by plasma membrane and gram-negative cell wall. It has multiple periplasmic flagellas which is located between cell walls and outer sheath. The morphology and motility of the organism can be observed in dark ground or phase contrast microscope. Negative staining, silver impregnation method, Fontana's method and Levidity's methods are used. Next, coming to the cultivation, resistance and antigenic structure. Cultivation. It will be having two kinds of organism, non-pathogenic treponemes and pathogenic treponemes. Non-pathogenic treponemes are also called as Reiter strain. It would be cultivable under strictly anaerobic condition. The medium used is Smith-Noguchi medium or digest broth. Pathogenic treponemes are also called as Nichols strain. It would be cultivable under complex anaerobic medium. The medium used is Nelson medium. It would be maintained in rabbit testis and would be done by serial passage. In Nelson medium, it is maintained for 1 to 6 days. Resistance. It would be inactivated by drying or by heating at 41 to 42 degrees Celsius for 1 hour. The organism would be killed in 1 to 3 days at 0 to 4 degrees Celsius. Transfusion syphilis can be prevented by storing the blood for 4 days in refrigerator. The organism can also be inactivated by the contact with oxygen, distilled water, soap, arsenicals, common antiseptics and antibiotics. The antigenic structure, it would be having three types of antibodies. The first one is reagent antibody. The second one is group specific protein antigen. And the third one is species specific polysaccharide antigen. Coming to the pathogenicity, the transmission would be through sexual contact. It would be entering through breaks in the skin or by penetrating through mucous membrane. Clinical disease sets in after an incubation period of 10 to 90 days. The organism would be entering through sexual contact and the organism would be penetrating the intact mucous membrane or through breaks in the skin. It would invade the bloodstream and spreads to other body sites. It would cause endarteritis and progressive disease tissue destruction. Syphilis disease would be occurring in three major stages: primary syphilis, secondary syphilis and tertiary syphilis. Primary syphilis will exhibit symptoms like painless indurated hard ulcers. It is also known as chancers. Chancers are present on genitals, mouth or breasts. Rectal chancers would be present in male homosexuals and in females chancers present are on vagina and cervix. Secondary syphilis. It would be entering between 6 weeks and 6 months. It would be characterized by the appearance of red raised rashes on any part of the body, including palms and sole of the feet. And it also have pale moist papules which would be seen in the body. Tertiary syphilis. It would be characterized by granulomatous inflammatory lesions which would be present in liver, skin and bones. Cardiovascular lesions and degeneration of nervous system is also observed. Coming to the laboratory diagnosis. It consists of two major methods, microscopy and serological tests. Microscopy. Specimen should be first collected. It should be cleaned by soaking in warm saline. Wet films are made and cover slips are applied. It is viewed in dark field microscope and recognized by slender structure, regularity of spirals and slightly pointed ends. Direct fluorescent antibody test and silver impregnation test is used for the diagnosis. Coming to the serology method, non-treponemal tests or standard test for syphilis are the first one. This test uses cardiolipin. The antibodies present are reagent. These both, that is the cardiolipin and the reagent, would react together to give a flocculation, and through this we observe the results. Coming to the treponemal tests, treponemal organisms act as antigen to detect antibody formed. These tests are mainly used to confirm the results obtained from non-treponemal tests. Treponemal tests are of two types, those using cultivable treponemes and the second one, those using virulent treponeme pallidum. The tests coming under non-treponemal tests are Wassermann reaction, also known as CFT, Kahn flocculation test, VDRL test, RPR test. And the ones coming under treponemal tests are under pathogenic treponemes, it is T. pallidum immobilization test, T. pallidum agglutination test, T. pallidum immune adherence test, fluorescent treponemal antibody test, T. pallidum hemagglutination test, enzyme immunoassay. And under cultivable treponemes are Reiter protein CFT. This is all about Treponema pallidum. Thank you.